Ma'am, this is a matter of absolute joy and happiness for us to have you with us. And it will be very much educating and enlightening for us to listen to the session. And your topic is like so much in line with the current scenario that the whole globe is suffering in every facet, be it education, be it health, be it any facet, but everyone is facing trouble on this, right? So before we commence the session, I would like to introduce the chair for this session. And the chair for Dr. Roma Ghai, she is chairing this session. And she is presently working as an associate professor in the Department of Pharmacology with KIET School of Pharmacy. And she's with KIET School of Pharmacy for good 14 years, good 10, 15 years. She joined here in 2009, and she holds more than 15 years of teaching experience and have been guiding postgraduate students in their research projects. And apart from this, she's got 3 patents published to her credit. She is the author of 2 books, one entitled Pharmacological and Toxicological Screening for pharmacy students, and the other is the textbook of pharmacovigilance for bachelor students. And she has been delivering her training programs with the Department of Science and Technology, Government of India. And she has also developed a virtual experimentation lab for the university students in these pandemic times, so that the students can learn while still they are in the front of their computer screens. She, to her credit, have approximately 20 publications in national and international journals of repute, and has been presenting her research work in several seminars and conferences. And she has recently presented a paper in Endeavor Research Group, San Jose in Canada. And now I would request Dr. Roma Ghai to please welcome Dr. Vesna with a bouquet of flowers.
Greetings to all. Yeah, thank you so much, Himanshu. Yeah,
Yeah.
first of all, I would like to thank the organizers of Atal FTP for giving me this opportunity to chair the session. I consider it a great honor to welcome and introduce an eminent international keynote speaker of today's session, Dr. Vesna Rastija. I welcome you, ma'am, for your gracious presence as a keynote speaker and a very highly resourceful person of today's session. It's my proud privilege to introduce ma'am. Madam is an associate professor at the Faculty of Agrobiotechnical Sciences, Osijek, Croatia, teaching chemistry at the Underground University Study Program. She was born on 26 September 1970 in Osijek, Croatia. She graduated in biology and chemistry at the Faculty of Pedagogy. From 1995 to 1999, she worked as a teacher of biology and chemistry at university. Since 1997, she has been working at the Faculty of Agriculture in Osijek. She completed her PhD in 2007 in chemistry at the Faculty of Natural Sciences and Mathematics in Zagreb. She was the leader of 2 research projects, and on 2 research projects she has been associate. She is the author and co-author of 46 scientific papers coded in the Web of Science, H Index 13, and she has authored 2 chapters in the books. Madam has participated in numerous scientific conferences, and she is a member of Croatian Chemical Society and a member of editorial board of 2 scientific journals. Madam shall be presenting her talk on a very, very important topic, molecular docking of peptide derivatives as inhibitors of SARS-CoV-2 S protein. As we all know that the world is facing COVID-19 crisis, so I really think that this topic is the need of the hour, and we can learn a lot from ma'am. We are really honored to have a time from a busy schedule. So I again welcome Dr. Vesna Rastija ma'am and request ma'am to deliver her talk and enlighten our participants. Ma'am, please.
Thank you. Thank you very much. Thank you. Greeting to all. Thank you all for great opportunity to give me great opportunity to giving you my lecture about molecular docking and the fresh new results about the drug design of SARS against SARS-CoV-2 protein. Now I'll present you my, can you see my presentation?
Ma'am,
Can
now
you
you can
see?
share your presentation. You can share your presentation now.
Okay, okay. You cannot see? No?
Right.
Not now, but
Not now. Okay, okay, I have to share. Enter full screen.
Now see the top right corner, there will be a upward pointing arrow. Click on that arrow. You will find windows there.
Okay, can you see now presentation? No?
No,
no,
not right
not
now.
now.
No.
No.
Yeah. Ma'am, you have to click the arrow just the top of the this MST screen. Just the top.
Open share.
It's coming now. Yeah, yes, ma'am,
Okay.
it's visible.
Yes.
Now
And
gone
now?
again.
Now you can see?
Now
No.
it's gone.
No, it's gone again. Ma'am, if it is fine with you, you can share your presentation to us and we will control it from here for you. No issues in that.
Just right in the left side of your leave button for MS Teams, you could see an arrow, and through
Yes.
that you can upload your, you can share your screen or you can upload your PPT, PowerPoint
Okay,
presentation.
I'll open share. Share drive. Okay. Now you cannot see nothing. No?
No, not yet.
Oh. Open share.
Ma'am, I
don't
think you
know
just
why.
click the arrow.
Yes,
And
I am.
just click
I
the
Click
see
the
arrow.
arrow.
arrow,
Okay.
Yes,
And
just click it.
then
And
click on
In
the
that
windows.
you go in Windows.
Some number will be there in the bracket of the window. And in window, if your PPT is open in the background of your this MST screen, then it will be visible in that window. First, you need to open your PPT and
okay. Now can you see?
yes, now it's
yes,
coming.
yes.
Yeah.
Yes,
Now
Ah,
it's
yes.
up.
okay, nice.
Yes,
Okay,
it's
good,
come.
good. Okay,
Kindly put it in the full screen mode.
okay,
Thank you. Now it is visible.
okay. You will see. I don't know. Now it's okay.
Yes, ma'am. It is
Yes.
absolutely
Oh,
fine.
nice.
We can see.
Okay, good. I'm sorry for inconvenience. I hope you can see my presentation without problems. Okay. Uh, now I have to thank you all again for a great opportunity to give a lecture on your conference. Again, my name is Vesna Rastija. I'm a professor of chemistry on the Faculty of Agrobiotechnical Science in Osijek, Croatia. I hope that you are all well, and this lecture will be very interesting and useful for all of you. The title of lecture is Molecular Docking of Peptide Derivatives as Inhibitors of SARS-CoV-2 S protein. First, I have to say something about computational method in drug discovery design. Before we start to drug discovery design, we have to identify the target. Target is enzyme, protein against drugs has to be developed. Competition at the drug design consists of two parts: the structure-based competition at the drug design or ligand-based competition at the drug design. Structure-based competition at the drug design consists of target structure. First, we have to identify the target structure. Now we perform ligand docking. molecular docking, de novo design, molecular dynamic simulation, and pharmacophore modeling. Ligand-based computation at the drug design consists of the ligand structure and information, quantity structure activity relationship, pharmacophore modeling, and ligand-based virtual screening. After the leading components was identified, now we can perform the test of the leading components in vivo to identify future drug candidates. What is the molecular docking? Molecular docking is computational method in drug discovery. Purpose of molecular docking is to predict the preferring binding orientation of ligand, small molecules or drug candidates, to the receptor. Receptor could be enzyme, protein, carbohydrate, and nucleic acid when forming a stable complex. In this video, you can see the searching for preferred binding orientation during the molecular docking. The main objective of molecular docking is to, as I said before, to identify new lead candidates for drugs and evaluate interaction of ligands with the receptor. Docking tools can employ various search algorithms. It could be genetic algorithm, fragment-based algorithm, Monte Carlo algorithm, molecular dynamic algorithm, etc. Types of docking could be flexible, rigid body docking, and flexible docking. What is the flexible ligand docking? Flexible ligand docking incorporates target as a rigid molecule. It is the most commonly type of docking. Rigid body docking kept a rigid molecule both and ligand, both target and ligand molecules are kept as rigid molecules, and flexible docking involves both interaction molecules and ligand molecules as flexible. Software that we perform for the molecular docking could be various. It could be free available on the internet or you can purchase it. The most commonly software are listed here. We are using the plants, CellDoc, Autodoc, Vinam. I'm using a Egam doc software. You will see some examples with this docking software. Okay, now I will introduce you with Egam doc software. This is the free available docking software. You can freely upload it from the internet. Its fitness function using the van der Waals energy, electrostatic energy, and H bonds. And the docking scores are using a total energy with all three types of interactions. Egam doc molecular docking consists of a few steps. First steps is prepare the protein binding site. Next step is prepare the components library. Then we are perform molecular docking, and after all is followed by post-screening analysis. First, we have to identify the target. Target could be the protein, enzyme that is related with the illness that we have to examine. I'm using the protein data base, PDB base, searching entering the code of the enzyme of protein or the name of enzyme protein. We are looking for the complex with the ligands that fits the best with our compounds that we use. For this study, I'm using the crystal structure of COVID main protease in complex with inhibitor N3 or some other protein. When we open the Egam doc, first we have to prepare the binding sites. This is the interface for the Egam doc. Click on this button, we are uploaded the binding sites of the protein. We can retrain or not retrain the reference ligand. It is our decision. After that, we prepare the compounds. For this study, you will see I'm prepared the 19 compounds. Now, we have to set up molecular docking parameters. This is the population size, generations, number of solutions. One important, I have to say that if we enhance the number of population size, generations, and number of solutions, such as the number of compounds, the molecular docking will be very long. Okay. This is the run of docking screening. The software looks like this. And after the molecular docking is finished, it is followed the post-analysis. Egam doc generates the interactions profiles. You will see here the best docking poses, the values of total energy and values of van der Waals, H-bonds, electrostatic interactions. We can sort the compounds according the highest energy, binding energy, and perform the ranking of the binding poses. In the next picture, we can see the tables of interactions with amino acid residuals. And this is the results of molecular docking, the ranking of the best docking poses. After after we finish the molecular docking, we can make a visualization of the interactions of the best docking pose in the binding site of protein or enzyme. For this purpose, I'm using Discovery Studio Visualizer. This is the software also free, available on the internet. You can upload it and you can use it for the visualization. Also, you can use the very good program Hexa. This is not too much scientific, but it makes very nice figures of binding sites, a complex of ligands into the binding site of the receptor. Okay, now I will introduce you with COVID-19 illness. COVID-19 illness, we all know, unfortunately, this is the illness such as common cold or severe acute respiratory syndrome and the Middle East respiratory syndrome MERS. Common signs and symptoms can include fever, cough, tiredness, shortness of breath, of difficulty of breathing, muscle aches, headache, chest pain, pinky eye, vomiting, diarrhea, and the other signs. This is the fresh news from World Health Organization from 18 July this year. This is the cumulative numbers of cases reported globally is now over the 180 million people have COVID-19. The number of deaths exceeds 4 million people until now in whole Earth. This is the currently designed SARS-CoV-2 variants. We know the alpha, beta, gamma, and fresh new delta variants that is now also begin in Croatia also. And this is the world map of vaccination. This is the World Health Organization have information that the more than 8.85 billion doses have been administered across the 180 countries. But despite of efforts to extend its vaccination through the world, many countries across all 6 World Health Organization regions continue to experience surges in COVID-19 cases. Over the past week, highest number of new cases were reported from Indonesia, United Kingdom, and United States of America. We know all that there is no specific antiviral treatment against COVID-19, but this is not recently, but recommended combination of drugs are remdesivir, hydroxychloroquine, and azithromycin. Okay, now we will talk about SARS-CoV-2 and S protein. SARS-CoV-2, as I said before, is a severe acute respiratory syndrome coronavirus, which causes disease COVID-19. This is the picture of this this virus. SARS-CoV-2 consists of few structural proteins, the membrane glycoprotein, nucleocapsid protein, spike protein or S protein, and envelope protein E. We will focus now on the SARS spike surface glycoprotein or S protein. This is the picture of this protein. This protein plays an important role in mediating of viral infection. So because it is responsible for the viral recognition of angiotensin converting enzyme 2, ACE2. It binds with the human ACE2 and uses it to entry to invited target cell. This is the architecture of SARS-CoV-2 glycoprotein. It is the homotrimer in perfusion conformation, chromotrimer A, B, and C. It has N-terminus receptor binding domain, and this is the S-terminal domain 2, S-terminal domain 1. This is the central helix region, connector domain, haptid repeated 1, and the cleavage site between S and 1 domain. This is the architecture of SARS-CoV-2 glycoprotein. In a single protomer, you can see easily the architecture of this protein. We can see the S1 subunits and S2 subunits. This is the receptor binding domain, subdomain 1, subdomain 2, and terminal domain, haptid repeated domain, central helix, it's a central helix domain, and connecting region. What is the mechanism of action of S protein? S protein, S1 subunits, binds to the hosted cell receptor through its receptor binding domain. Okay, this is the receptor binding domain. After that, proteolytic cleavage is happened occur between the S1 and S2 cleavage site. Two heptad repeated regions form 6 helix bundle structure, which triggering the release of fusion peptide. S2 subunits is responsible for the fusion of the viral and cellular membrane. What is the possible drug against SARS-CoV-2? Possible drugs could be the inhibitors that binds to the receptor binding domain, which induce the conformational changes in S-glycoprotein and prevents the binding to the ACE2 of human. So, for this purpose, we have used the potential inhibitors. It is peptide type compounds, which previously was proven as chymotrypsin-like protease inhibitors. We performed the molecular docking on 62 optimized structure peptide type compounds and antiviral drug remdesivir. The three-dimensional structure of SARS-CoV-2 spike glycoprotein in complex with N-acetylglucosamine we uploaded from the, as I said before, from protein data bank. It is how it looks. This is the protein data bank. If you enter the code for the 6VSB, you can find the you can find the downloaded the structure of S-glycoprotein with the ligand, with this ligand N-acetylglucosamine. After performing molecular docking, we find that the highest binding affinity toward S protein has compounds coded 21. This is its structure. This is the energy of binding. This is the docking scores of the best docking pose of peptide type inhibitors and standard antiviral drug remdesivir in complex with SARS-CoV-2 spike glycoprotein. As we see, the 1, 2, 3, 4, 5, 6, 7, 8, 9 compounds had better energy of binding than standard drug remdesivir. Compounds 47, which exhibited highest inhibitory against proteases, showed lower binding affinity to the protein S protein. And antiviral drug remdesivir used for the treatment of COVID-19 demonstrate low binding affinity. So we can conclude that these dipeptide type compounds could be the potential drug against the COVID. And so, if we look these compounds 47 and its binding to the SARS-CoV-2 spike protein, we can see that it creates interaction with both subunits, subunits 1 and subunit 2, generating the H bonds with lysine 310 and threonine 604. Next compounds, 33, you can see he is ranking on this place. Compounds 33 is binding, creates interaction also between both subunits, subunits 1 and subunits 2, generating the H bonds, more H bonds with lysine 310, threonine 604, threonine 602, threonine 603, and threonines 941. And the best compounds, compounds 21, had highest binding affinity and it is binding only on the one subunit. This is the subunits 1, forming the cluster of H bonds and which allows the stronger binding to the S1 subunits. So this is the all three compounds picture of all three compounds. The red one is compound 47, blue one is compound 33, and yellow compound is compound 21, which exhibited the highest binding energy. And as we see, the compound 21 tightly fits to the subunits 1, while the other two compounds tightly fits to the both subunits. Compound 21, making the interaction with only one subunit, prevent binding the S protein to the human ACE2. So in conclusion, we can conclude that molecular docking helps us to screen the potential therapeutic targets against corona-19. And tested compounds have ability to bind on the receptor binding domain of spike surface glycoprotein virus, and thus may inhibit the binding to the host cell membrane. This is the references. This is our two new paper about SARS-CoV-2 drug design. We made the paper about structure features of peptide type compounds with COVID main protease inhibitors, and identification of food compounds as inhibitors of SARS-CoV-2 main protease using molecular docking and molecular dynamic simulation. Thank you all for your attention. I hope that you find this lecture interesting.
Yes, any questions anyone have regarding the topic? Please ask your queries.
Ma'am?
Yes, both, sir.
Can I ask?
Yes, sir.
Ma'am, it's regarding this particular antiretroviral drugs that we are applying. So, what, which receptor you think they are actually working in this particular case?
Receptor?
Yes, yes.
Receptor was S-glycoprotein. As we see in presentation, it was the protein in the surface of COVID-19 virus.
If there is any particular mutation is there, that is we are seeing very several variants of this COVID are coming, recent being the Delta plus variant. So if there's particular mutations are happening, so what are the chances this particular receptors will also get modified? If there is any change in the protein structure, then will these antiviral drugs also work if there is any mutational change in that protein receptor structure?
I have no information of differences between the variants in SARS S protein. This is not my area, but I think that it is the differences between S protein and mechanism of action between different variants. It could be, maybe it is not very great difference, but it could be, because we know that the last variant, the Delta variant of SARS-CoV-2 protein is very infections, and it is infections is possible infections is because of different structure of S protein. It is possible, yes.
One last question, ma'am, may I?
Yes.
Can I ask something regarding vaccination or vaccines?
Vaccine. Okay, about
Ma'am,
yes, for vaccines I know only that vaccines is messenger RNA that initialize the generation of S protein in
my
our immune
question is
system.
actually, madam, regarding the vaccine. So we are having several. You are done this vast research on different proteins present on this particular virus. Actually, that I can understand from your presentation. So, ma'am, my question is the sum of the vaccines that are available that are whole inactivated virions. Some vaccines are particular subunit part or proteins part. So if mutation is occurring, suppose
Yes.
I am injecting the whole this whole attenuated vaccine, whole agent attenuated vaccine. So I don't know from which part or which fragment of that virus will be used by the T helper cells to produce a antibody. So, which kind of
Yes.
vaccines do you think will work better in case of mutations? The whole inactivate agent, or a particular subunit, or a particular this particular protein, RNA-based particular protein, or any subunit vaccine?
Yes, I think that you thinking very good and good right. Uh, I don't know this vaccine are generate that thinking about the structure of S protein and S protein uh, S protein really I think that that will be great problem, you know. So because we see that some vaccines doesn't have an answer on the new Delta variant of virus. This is the possible explanation why some vaccines doesn't have answer, and the people who vaccinated also have illness had COVID-19. Yes.
Hello, ma'am. I have a question. May I ask?
Okay. Yes.
Yeah, first of all, ma'am, thank you so much for such an illustrative presentation. My question is that there were few reports few days back that extensive use of remdesivir has led to the emergence of variants of this COVID virus. So, what is your take on this, and can you enlighten us on this?
Okay. I have also one paper about using the remdesivir against the COVID-19. And they found some interesting information. You cannot use it remdesivir without using the zinc drug. Without zinc, remdesivir doesn't act.
Okay.
Zinc and remdesivir you have to use together.
Zinc
So
is azithromycin, if I'm not wrong.
zinc,
Uh,
metal, iron, zinc
Okay,
iron.
zinc. Okay, I'm sorry. Sorry.
Zinc iron, yes,
Okay,
because
zinc.
it is very important for the interaction with virus. And also, you know that in some country they are using, as you see in my presentation, with free medicine. In Croatia, uh, some doctors are prescribed azithromycin as a drug against COVID, and
Yes,
it was shown as useful drug.
yes. In India, it
At
has
the
been used
first
extensively.
symptom
Yes,
of the pneumonia, some physicians prescribed the azithromycin. It was very useful because this COVID-19 did not was presenting in very in the immediate variant. Okay.
okay, okay, ma'am. I guess we have one question from Miss Devlina Tiglani, ma'am. You may unmute yourself and you may ask your question.
Yes, sir. Ma'am, for your presentation, it was really knowledgeable.
Ma'am, you're not audible. You're not audible.
Sir, am I not audible right?
No, ma'am, we can't hear you. We can't hear you. Sorry. There is way too much background noise.
Sir, am I
Please
audible
type
now?
your. Yes, yes.
Yes, now you're a bit audible.
Thank you, sir. And thank you, ma'am, for your presentation. Actually, first question that I want to ask that recently you have told us that remdesivir should be used with zinc. Can I know the reason why? Why is it necessary that remdesivir should be used with zinc or iron? Yeah, why it should be? I'm asking why it should be used with zinc for the
Okay. I thank you very much for your question, and I hear you. That took so good, but I hear you. I think it's very important because it is, yeah, it is need to be during the interaction of remdesivir with the binding sites. Is a mediator of these interactions. Okay.
And ma'am, my second question is you have selected the compound or the protein named V6VSB, right?
I cannot hear. Sorry.
Ma'am, you have selected the protein named 6VSB, which is binded with N-acetyl D-glucosamine. Why it is selected? Because most of the time we use the compound that is binded with the most active drug, and recently we have heard that it is remdesivir. So why glucosamine binded drug or protein?
I'm sorry, I can hear
Uh,
your
Ma'am,
question
your voice is not clear. We can't hear the question
because there are
properly.
lots of
Yes, there is too
voices
much
behind.
background noise.
Yes,
Ma'am,
Sorry
you
for
can
that,
behind.
please
sir.
type your question in our WhatsApp group also. Then we can ask Madam from there.
Sir, the WhatsApp group is only available for the admins to type, not
In the
for us.
conversation group, in the conversation chat, you can put your question. Then we will ask on behalf of you.
Yes, you can put the question in your chat, and I will.
Ma'am, you can now you can send the question in the WhatsApp now.
Okay.
Okay,
You
sir.
can send the option in the WhatsApp now.
Any participant who is having any question regarding the presentation, so you can direct those questions to Dr. Vesna, and she would be answering those questions.
Can I ask one question, Dr. Vesna?
Yes.
Yeah. Actually, I just want to know that there are so many variants coming in this coronavirus. It's like alpha, beta, delta.
Yes.
Delta plus is also there. So, is
Yes.
there any change in the structure of the proteins also? Is there?
Uh,
Yeah.
Yes, I think that you can find some small differences in the genes of this, making the genes of these proteins. As I show you the architecture of S protein,
Yes.
the small differences in the genes makes this so genes. So it is the this is the mutations makes on genes, and these mutations make different genes and different variant of this virus, unfortunately. So we think that the most of the people must make the vaccines, and the mutations will be finally stopped, and we finally finished with this illness.
Ma'am, one question from my end
Yes.
again. Ma'am, in particular UK, we are hearing about a vaccine cocktail. Some particular vaccine cocktails they are preparing by mixing different subunits of different variants. Hello. So,
I'm
ma'am, can one person take so full? Suppose for a particular vaccine, we need to take 2 doses. Okay, so after taking two.
Ghosh sir, unmute yourself. Ghosh sir, unmute yourself. Host sir, you unmute yourself.
Can you hear me?
Yes, ma'am, we can hear you.
Okay, so I'm sorry, colleague, I didn't hear your last question very well. I think you're asking about the number of vaccines. I have no information how the vaccines act
Ma'am,
Hello,
against.
am I audible? Am I audible?
yes.
Yes, please, can
Yes,
Am
sir,
I,
you
you
no,
are
no,
audible now.
repeat your questions?
Actually, I am repeating my question. Actually, my
Yes,
question is that in this mutation, the protein structure is getting changed.
yes,
Suppose there are several vaccines now in the markets from Sputnik V, AstraZeneca, Moderna, Pfizer.
yes,
Suppose I am starting with this AstraZeneca Covishield. I have taken if I am taking the 2 doses. So
yes.
after certain gap of 3 or 6 months, can I take another vaccine
Um,
like Covaxin or Sputnik for better chance, better
yes.
immunity?
Uh, really, it's a good question. I also have the same question to my. I taking also myself. I took the 2 doses of Pfizer. It was very hard after that. 12 hours after that, I was very bad shape. But and after that, I didn't know. If I took the AstraZeneca after 9 months, really, I don't know. Maybe it could be create new mutation. I really don't know answer of this on this question, but it is very interesting to think about that and study.
Thank you.
Thank you.
Ma'am, ma'am,
Yes.
the question of the participant, Dr. Devlina, she has sent the question via WhatsApp. I'm reading that question to you now.
Okay.
That most of the times we select the drug that is bind with highly, sorry, just a second.
Mm-hmm.
Most of the times we select the protein that is bound to the most active part of the drug, but you have selected the protein that bound to NAG. So any
Yes. Binding
specific
energy.
reason behind that?
Sorry, I didn't heard the
And
last question.
I'm saying her question is that most of the times we select the protein bound that is we select the protein that is bound most actively with the drug, but you have selected a protein that was bound with NAG. So any specific reason behind that?
In molecular docking, when we treated the best drug is the best leading compounds is the compounds that makes the highest, they release the highest binding energy. So it makes more stable, most the stable complex with the receptor. This is the main methods in molecular docking. If you understand your questions, this is the simple answer.
And can you elaborate it a little more? Ma'am, can you elaborate it a little more?
Dr. Vesna, she wants the answer in a more simplified terms.
In more elaborated way, she is asking. So, ma'am, if there are no further more questions, can we ask the chair to conclude?
Yes, Himanshu, should I conclude?
Ma'am, that's what I'm asking. Dr. Vaishali? Ma'am?
Ma'am, should we conclude?
So now I would ask the chair, Dr. Roma Ghai, to conclude the session by Dr. Vesna. Thank you.
Thank you, Himanshu. Thank you, Dr. Vesna ma'am, for a wonderful and highly informative and valuable presentation and very interesting too. Thank you, madam, for sharing your profound knowledge on the topic of blocking of peptide derivatives as inhibitors of SARS-CoV-2 S protein in a very practical and crystal clear manner. We have learned steps, many steps, like you made us understand the computational methods in drug discovery design. You told us about docking tools. You told us the steps of docking, like preparing binding sites, preparing compounds, and doing docking and doing the analysis. You've showed us the actual implementation of all these steps in a very crystal clear manner using IgM-doc, and how peptide and chymotrypsin inhibitors, they can bind to the receptor binding domain of the SARS-CoV-2 S protein, and they can induce the changes in S glycoprotein, which can actually act as inhibitors. So it was indeed a very highly knowledgeable session, ma'am. I'm really sure that our participants must have benefited from the session. Thank you so much, and we are very grateful to you for sparing your valuable time with us.
Thank you.
Thank you so much.
Thank you. Thank you all.
Thank you, Vesna. And
Hello.
see you again in the next
Thank
events
you.
or for
And,
further collaborations.
yes.
Thank you.
Ma'am, and as a token of gratitude, I would request the chair to please present a certificate to Dr. Vesna. Everything is happening in this virtual world, so please.
On behalf of KIET School of Pharmacy, I, Dr. Roma Ghai, present you this certificate of appreciation as a token of respect and gratitude. Thank you.
Thank you very much. Thank you. Thank you all, and goodbye.
Goodbye. Thank you.
Goodbye.
Thank you, ma'am. Thank you.
See you again, ma'am. See you again.
See you.
Nice to hear from you.
Bye.
Thank you. So, dear participants, now I would be sharing the attendance as well as the feedback link. So, you are requested to fill the feedback as well as the attendance for the session. For attendance for the day, both the links will be shared in the WhatsApp, and you are requested to fill it by 4 p.m. Later that the forms will close and we cannot manually override them. So please fill the attendance and feedback by 4 p.m. positively. Post that we would not be in a state to entertain any requests. Thank you so much. Shali ma'am, if it is okay, so can we conclude the day?
Yeah.
Please, sir.
Thank you, Vishali
Thank you
ma'am.
so
Thank you,
much.
Himanshu, and all the
Thank you,
organizers.
sir, and thank you all the participants also.
Vishali
Sir, you can conclude. Yes,
Vaishali
sir.
ma'am, kindly put this time limit in the WhatsApp also, because some may or me
No,
tell
we
that they
will
were
put that in the WhatsApp also, sir.
Sir,
Thank
add kar rahe hain.
And so thank you everyone. We will meet tomorrow, and Dr. Praveen will be sharing the schedule for tomorrow's technical sessions via email and WhatsApp. And Praveen sir, I think you were saying something. Please continue, sir.
Yeah. Dear participants, I would like to say, please keep on checking your emails on daily basis so that you will get the further informations and schedule for the next day. Thank you very much. Thank you, sir.
Thank you.